Okay, guys, just to wrap up our discussion on biotechnology, we really should know about PCR. And I'm going to describe it here, but as you already know, there is a quiz coming up in lab eventually, and that also has PCR in it. So I'm going to probably ask a lot more about PCR in the lab quiz than I do in the lecture exam. But it will show up in both places, okay? PCR, polymerase chain reaction. So here we are now. We're taking in all of the information that we understand about DNA replication and how it is that the DNA polymerase molecule works. And we're going to apply that to a piece of DNA. Now, it's very hard to study DNA if there's not enough of it, right? If you don't have enough DNA, then it's really hard to study and sequence that DNA and figure out what it is you're working with. So polymerase chain reaction allows us to do just that. It allows us to amplify a certain segment of the DNA that we're most interested in, not just all the DNA, but the portions that we're most interested in, to then determine what's there, study the gene if it happens to be a gene that we're studying, or just to identify the existence of a pathogen or of a bacterium, because it might be a sequence that is unique to that pathogen or bacterium. So let's start with something real simple. We have a target DNA. We have a piece of DNA. We need to amplify it. It could very well be a sample from a crime scene, let's say, right? So you have a hair follicle. And in that hair follicle, there's going to be some living cells. And those cells will have intact DNA. And I want to tell if that hair belongs to someone, right? So what we would first need to do is take a sample, isolate the DNA, and I would need to heat to denature that particular set of DNA molecules. Now, when we denature, we're going to break hydrogen bonds, right? So we're going to break hydrogen bonds. When it's a restriction enzyme, it does it in their sticky ends. In this case, we're going to break the hydrogen bonds from both strands, right? So the strands are going to come apart, okay? These anti-parallel strands will denature at a high heat, okay? Now, the next thing we need to do is to introduce the appropriate tools to act upon those single strands. We would want to add the three following things. Primers. Now, these would be small sequences of DNA that are single-stranded, and those particular primers would be specific for only certain DNA within the target DNA we're working with. So you have to know something about the DNA that you have isolated in order to do PCR. If I've isolated a piece of DNA and I want to just amplify the portion of that DNA that is the insulin gene, or just amplify the portion of that DNA that is some other enzyme, or it doesn't even have to be a coding region, right? If it's a eukaryotic cell, there could be non-coding regions I could also target. But we do need a specific sequence of nucleotides and a small primer in order to do this. We would also add a mix of nucleotides. For this, we would need A, C's, T's, and G's, right? So we're going to have DCTP, DATP, DTTP, and DGTP. So these are our A's, C's, T's, and G's, right? So these are our nucleotides, and we're going to use that to make more DNA. And then finally, we need a DNA polymerase, and not just any DNA polymerase. We use Taq polymerase. Now this stands for, the capital letter stands for the genus, which is a thermophile. So it makes sense you'd have T for that particular letter. And then Aquaticus is the species name, okay? So the DNA polymerase that we're using is actually from a thermophile. This is an enzyme, this polymerase, that will not denature in the presence of heat. So it's okay to have the Taq polymerase, this enzyme, in a tube where we introduce heat. So here we have our DNA polymerase. Now we know that it's actually Taq polymerase from a certain thermophile. After we heat the DNA and we have the strands apart, now what we would find would happen is the primers would actually attach to the target DNA. Okay, so we'd cool it enough for these hydrogen bonds to form, and these hydrogen bonds would form between the primers and the target DNA, but only where that primer sequence matches up with the sequence in the target DNA. And the same thing would happen with our other primer, right? And it's going to work this way on this complementary strand. It's going to work this way on this complementary strand, and we're going to double the amount of that particular sequence. Now, after this has occurred, I can heat the whole thing up again, okay? So now I've got 2 pieces of DNA, and I heat it up, and I have 4 single-stranded DNA, and we'd have our primers bind, our DNA polymerase do its thing, and this would just happen again and again and again and again. Now this very quickly gets big, right? If we take 1 piece, it becomes 2, 2 becomes 4, and if we do this cycle like 30 times, this heat, cool, heat, cool, heat, cool, right? We're going to get tons of DNA of just that one segment that we're looking for. Now, if that one segment is only found in one particular, let's say we're not looking at hair follicles anymore, but I want to look at a fecal sample and I want to run PCR to look for just a sequence that would only be found in C. diff, or just a sequence that would be only found in some other pathogen. We could do that, right? You wouldn't even need to culture the organism. All you would need to do is to run a PCR on that particular sequence that's only found in C. diff. Now, just as well, we could use PCR, and of course we are today with the coronavirus, right? We're taking a sample, okay, from way back behind the nose, right, the pharynx. We're taking that sample with some mucus on it, and we're looking to see, is the virus in that sample? We run PCR, we find out if there is the presence of that sequence that we only know to be associated with coronavirus. So this is a powerful tool. What are we using? Our knowledge of how it is that DNA polymerase works. We're using our knowledge of a little bit of sequence that we know to be on the target, or we are looking for on the target, and it's really a powerful tool. Okay. So what are some of the things that we do with this particular tool of polymerase chain reaction, PCR? This is used to clone DNA for recombination, amplify DNA to detectable levels, like maybe that crime scene example, or maybe some scientists have done this with woolly mammoth DNA. You might have seen it in the news over the last few years. They found that some DNA in samples, and they've been able to use PCR to amplify those samples of DNA to detectable levels, and then use that to figure out how related they are to modern day elephants. We can sequence the DNA. Of course, we've done that. Now we know the entire sequence of the human genome. Again, we owe a lot of that to this early development of PCR. We could diagnose genetic disease, so I could take a human cell and look for a particular sequence that's known to be associated with lots of genetic diseases. And we can detect pathogens from samples as well, like the C. diff example or the coronavirus example that I mentioned. The applications of PCR, there are enormous. There are therapeutic applications where we can genetically engineer human proteins to help people. Vaccines are produced partly by using PCR. Gene therapy. There's the Genome Project I already mentioned. There's PCR has been used to further our basic understanding of DNA and even to fingerprint individuals to see who is related to who, for example. We have genetic testing. We even have agricultural applications where we can modify plants, like I mentioned, GMOs, right? We can modify plants so that they are able to withstand certain environmental pressures, whether it be heat or cold, or it's too dry or it's too wet. Pests, right, that would otherwise infect the plants. A lot of these kinds of changes can help with that. And so here are a few, I don't expect you at all to memorize these things, but maybe you recognize some of these. There's a lot of agriculturally important products genetic engineering gives us. And this is all coming from our basic knowledge of recombinant DNA, use of PCR, and lots of other techniques. Now, one interesting example that I'll bring up here is there's a lot of good you can do, right, with biotechnology. You can make a rice, like this particular rice was made by Monsanto Corporation, and they were able to make this rice over here on the right, which has a much higher nutritive value than the rice on the left. Now this is all sounds good, right? And again, this brings me back to the science for the use of knowing and understanding things is not a bad thing. How we apply it is also important, though, culturally. If we apply our knowledge to feed more people, that sounds like a good thing. It so happens that this particular company also genetically designed their rice not only to be more nutritive, but to be sterile. So anyone who buys their rice cannot resow the seed from the rice. Okay, so you might argue that particular move to make the rice sterile is not a particularly ethical move. So you could make decisions, but that doesn't mean that our ability to genetically change the rice is necessarily a bad thing. Okay. Here are lots of human products that can be produced. Some of these you might have heard of before, like human growth hormone. People hear of that, I think, where people don't make enough growth hormone. Insulin, for sure, you've heard of this. And again, remember, these are all, most of the time, genes that are introduced into E. coli. E. coli, then like a horse on a farm, it's doing the work, right? It's churning out the product you want. And then you just isolate that product. Factor 8, I had a student 3 years ago that had a little nephew with hemophilia. So they were lacking a certain clotting factor. We can make that kind of thing. Just a long, long list here. And again, I'm not gonna expect you to memorize all of these, but I just want you to know that these kinds of things can be produced as a result of genetic engineering, which grew out of our knowledge of basic genetics with bacteria. Now, the last thing I'll mention here, and this one is just really hot in the news over the last 10 years or so, and that is CRISPR. Okay? CRISPR is a really powerful new technique that we now have in our toolbox. And let me just find here, I'll tell you sometimes I forget the precise letters here. So clustered regularly interspaced short palindromic repeats. Don't worry, I'm not going to test you on it, but you might want to look it up just to see what all of the big deal is about CRISPR. Okay, What I'd like you to know about CRISPR, the CRISPR pathway, is that this is a technique whereby bacteria can precisely cut invading DNA. Now this sounds like nucleases, right? Like a nuclease, it will precisely cut introduced DNA, like from a virus. But not only is it capable of doing that, but it's capable of taking the pieces of DNA from that virus and inserting them precisely into its own genome. Now that doesn't sound too unfamiliar either, because we know that recombination occurs every now and then. This is pretty common actually. But then what the bacterium does is it uses that sequence to recognize any future sequences of the sort. So it's almost like an adaptive immunity almost that these bacteria have designed for themselves, that they can recognize an old viral sequence. And as soon as that old viral sequence shows up, this CRISPR pathway will break up that virus very, very quickly. Now what we've done is we've harnessed this CRISPR set of proteins to utilize that for cutting DNA precisely the way we want and introducing new segments of DNA in those particular locations. So it's not just like a find function in Microsoft Word, right? I can find everywhere the word "the" comes up in a paragraph. This particular CRISPR pathway will find the sequence and interchange sequences in that location, okay? So what we've done is we've used CRISPR as a kind of find and replace mechanism in molecular biology today. We can use CRISPR to simply cut and inactivate genes, but we can also use CRISPR to cut in specific places and insert new functional genes that we want to be there. So imagine that. Imagine if you could find the sickle cell gene where there's this nucleotide mutation, right? Find it with CRISPR, remove it, and replace it with a normal hemoglobin gene. Or for another example would be cystic fibrosis. It's caused by a single nucleotide change. What if I could find all of those particular sequences where we had a mutated gene and replace it with one that is normal? This is the power that CRISPR might be able to give us. And its power is a little bit unnerving, actually. But we've come a long way with recombinant DNA since the 1970s when we were really just first starting with E. coli. So now we find ourselves kind of wondering, okay, what kind of rules do we put on what we're able to do? What kind of policies do we place around this powerful technique, CRISPR, that we now know about? Okay? So I think this is a good place to stop. There's a lot of stuff. I mean, you could have a whole course on biotechnology. I know we have one individual in our class that is a biotech major. So I'm sorry to him and to anyone else who finds this really interesting, but we have more to do with some other topics, and antibiotics will come up very shortly. Okay? Also part of Module 3, by the way, so be looking out for antibiotics next.